Okay, I see everyone loading into the chat room and the webinar. We'll wait just about 30 seconds or so, let everybody get loaded in and settled down, and then we'll go ahead and get started. Wow, huge turnout today. All right, it looks like it's leveled off, so we will go ahead and get started. Good afternoon, good morning, and good evening to everybody, depending on where you may be logging in from today. I am Rob Burgess, and I head up business development for SinoBiogical. And it's my pleasure and honor to welcome you all to the next installment in the SinoBiogical series called BioTalk Tuesdays, where we have cutting-edge seminars from world-renowned researchers in very unique and cutting-edge technologies affecting therapeutics, diagnostics, and preclinical basic research. So welcome to everybody. I also want to introduce Ms. Jane Hall, who is the Senior Marketing Manager for GenoBioTX, and GenoBioTX is co-hosting and co-sponsoring this webinar today, and they will be for the next 3 or 4 webinars, I believe. GenoBioTX is also known as Shanghai Model Organisms, and I know that our speaker has a special relationship with that company. So welcome, Jane. One housekeeping issue I want to bring up is if you have a question for our speaker, please put it into the chat box, and we will withhold all questions until the end of the webinar, and then I will walk through as many questions as I can verbally with our speaker. One other thing is, and I think this is in the chat box, but please send a quick note and tell us where you're from. We always love to hear how international our audience is and the makeup is of our audience. And so with that, it's my pleasure to introduce to everybody our speaker for today. It's Dr. Yi Zhu, who is an assistant professor in the Department of Physiology and Biomedical Engineering at the Mayo Clinic. Dr. Zhu's research is dedicated to understanding how senescent cells contribute to age-related diseases and neurological disorders. Dr. Zhu's major accomplishments include identifying the first generation of Senolytics and demonstrating potential to combat age-related dysfunction. Her current research focuses on creating new senotherapeutics targeting senescent glial cells, examining the effects of peripheral senescent cells on brain aging and Alzheimer's disease, and employing spatial omics techniques to chart senescent cells in human disease tissues. Dr. Zhu graduated with a BS from Beijing Normal University. She earned her doctorate from the University of Michigan, and finally she completed her postdoctoral training under Dr. James Kirkland at the Mayo Clinic. So Dr. Zhu, it is an honor to welcome you today to give our seminar, and now we will turn the screen over for you to share your presentation.
Thank everybody taking time to join the webinar. And first of all, thank Bob for the nice introduction. And my name is Yi Zhu. I'm assistant professor at the Department of Physiology and Biomedical Engineering. And I'm also faculty at the Kauka Center on Aging at Mayo Clinic. So today I will talk about my research on cellular senescence and senolytics in aging and diseases. So first of all, I want to disclose I am one of the co-inventors on Senolytics patents held by Mayo. And so here's an overview of my talk today. I will briefly introduce about aging and why we study aging, why we focus on cellular senescence, and then I will talk about more examples why cellular senescence can serve as a key therapeutic target treating age-related diseases. So then from this figure, it's obvious that aging is a greatest and also common risk factor for developing all kinds of chronic diseases. This is also telling us that it is how important to understand the aging mechanism. And also the aging mechanism will help us to understand those diseases, try to find a way to prevent or intervene those age-related diseases. And so with the decades of aging research, we pretty much now well established several hallmarks of aging. And obviously, cellular senescence is one of the aging hallmarks. And to be qualified as aging hallmark, first of all, is definitely associated with age. And also, more importantly, they serve as an aging modifier. Basically, it means that when you accelerate or deaccelerate this pillar, you're able to modify aging process. And what is a cellular senescence? So cellular senescence is basically is a cellular state. So when healthy normal cells receive stresses from either internally or externally, such as like replicative stresses, oxidative stresses, or UV lights, or chemotherapeutic drugs, they will respond to those stresses. First, they will have a cell cycle growth arrest, accompanied with increase of two commonly used marker for senescence are P16 and P21. They are basically cell cycle inhibitors, and also there is a so also there is a combined with dysfunction in the lysosome. We commonly use this to detect the senescent cells, which we call assay beta-gal. And there is a decline of the nuclear envelope and displayed by loose lamin B1, and the chromatin from nuclear and or from dysfunctional mitochondria. They will release from either mitochondria or nuclear to the cytoplasma, and then the cytoplasma, the sequential signalings sensed those fragments to induce a sequential subsequent events, which we call the senescence associated secretory phenotype, is a whole bunch of factors. They are bioactive. They include like lipids or enzymes or uh, uh, pro-inflammatory factors. And so once senescent, so uh, we know that senescent cell, the main function of senescent cell are carried by the SASP, but they also have normal physiological or beneficial effects, such as in tumor suppression, help wound healing, and also involved in embryonic development. However, with aging or disease, there is accumulation of senescent cells, and accompanied with increase of SASP. Once the SASP increase pass to certain threshold, they can be pathogenic to induce all kinds of diseases. And how they induce diseases, as you can see from this flow, like when we get stress from either internally, like a diet or chemotherapy, or outside stresses, there is accumulation of senescent cells, and with a increase of their SASP in the local tissue, they can induce local inflammation, and by secreting those enzymes or tissue distracting factors, they disrupting tissue architectures, impair their regenerative capacity. Overall, they will induce eventually lead to geriatric syndromes such as cognitive impairment, frailty, and also all kinds of chronic diseases. Overall, accumulation of senescent cells decrease resilience and decrease survival. However, if we stop senescent cell accumulation at this point, either by removing some senescent cells or inhibiting their SASP, we might be able to find a promising way to prevent or treat those diseases. And then let's talk about targeting senescent cell can enhance healthy lifespan. So as I said, to qualify to be aging pillar, this pillar need to be associated with age. From this heat map, you can see in both naturally aged mice or progenitor mice, there is accumulation of two senescence markers, also commonly used markers P16 and P21 across multiple organs. Not only in mice, also in humans. Now we know senescent cells accumulate in all kinds of tissues in humans as well. Then, back many years ago in 2009 and 2016, there is a collaboration between Dr. James Kirkland lab and Dr. Jan van Dersen's lab at Mayo Clinic. They co-invented this famous genetic proof of the concept study by by engineering this transgene under P16 promoter. So as you can see here, those are monomer of caspase 8. Once you gave the drug to AP201A7 to dimerize of those monomer caspase 8, you're able to induce apoptosis specifically in P16 high senescent cells. And by making transgenic mice, and then they published in 2009 and 2016 in both progenitor mice and naturally aged mice, they found that by specific induced apoptosis in P16 positive cells, they were able to increase or enhance healthy lifespan in mice. However, this strategy is kind of proof of the concept. This is not be able to translate to humans. So then we set out to test whether we can find a small compound to target senescent cells. So then back a few years ago when I joined my mentor, Dr. James Kirkland lab at Mayo Clinic, I set out to test whether we can find Achilles heels of senescent cells, which means senescent cells might rely on some pathways to be survived in the harsh condition. And through the heat map, so we also did microarray to find the transcriptome between senescent and versus non-senescent cells. As you can see from the PCR analysis and the heat map of the differential gene expression, they have so senescent cells has very distinct transcriptome. Then when I further looking into the transcriptome which are related to apoptosis or pro-survival pathways with the help of our bioinformatics support, we generated this network. Now we call this is senescence associated pro-survival pathway. So we speculate senescent cells rely on this pro-survival network to be survived through the harsh condition during aging or diseases. So then we set out to test whether we can target the network to specifically kill senescent cells. And indeed, when I knocked down the keynotes of the network, we were able to selectively kill senescent pre-adipocytes and some senescent HUVECs. And then the next step is that to screen the compound to target those keynotes or pathways. So after screening more than 50 compounds, we identified dasatinib, which inhibit PI3K delta, PI1, and P21, and quercetin inhibit also PI3K delta and PI1, and they both they both can be senolytics. And as you can see here, we first set out to test the in vitro assay, the cell survival assay. We found that dasatinib can selectively kill senescent pre-adipocytes, but not HUVECs. However, quercetin can be senolytics in HUVECs, but not in pre-adipocytes. So to enhance the efficacy of senolytic activity, we decided to combine dasatinib and quercetin. And next, we tested the in vivo effects of the combination of the senolytics drug, the combination of dasatinib and quercetin. And indeed, when mice were transplanted with senescent cells, they also carry a luciferase signal, as you can see here. So after the treatment, D plus Q, we see significant reduction of luciferase signal. So then we know that those senolytics are killing senescent cells in vivo. And then in the next study, the published from Dr. Kirkland's lab, they found that the naturally aged mice after treatment of D plus Q can significantly extend lifespan of those aged mice. And then also, when I look into the pro-survival pathways, I found there is one famous anti-apoptosis family, BCL2 family, popped up from the pro-survival pathways. And then I set out to test how BCL2 family changed during senescence. So I said BCL2 family BCL2 family members across 3 different cell types, so HUVEC, mRNA, and adipose tissue stem cell, like their predisposed. So we induce senescence by inducing DNA damage, and then we did a time course to assay BCL2 family members change. So we did find that BCL2 family member BCLXL, BCL2, and BCLW, they progressively increase during senescence process. And as you can see here, also same pattern as P16, and both in HUVEC and mRNA, however, not in predisposed. And the next question we ask whether inhibitor of BCL2 family can be senolytics. Indeed, when we tested in vitro assay by giving different dose of novatoclax, so the novatoclax is senolytics in both HUVEC and mRNA. However, and also not surprisingly, not senolytic in predisposed as we expected, because BCL2 family members did not change in senescent predisposed. So from this study, we started to notice that senolytics can be cell type specific or could be very context specific. And after we discovered a series senolytics, multiple independent labs tested the efficacy of senolytics in their preclinical mouse models for all kinds of diseases, such as obesity-induced metabolic dysfunctions, age-related bone loss, or liver steatosis, Alzheimer's models, and IPF models. So all kinds of preclinical models, they all showed very promising beneficial effects of the senolytics. So carry with us, the success of the preclinical testing. Of course, the next step is in humans. So the first human trial, the senolytics combination of dasatinib and quercetin were tested in IPF patients. So this first trial was published in eBioMedicine. They found that after 9 rounds of D plus Q, the physical activity was significantly improved in those patients. And also their pro-inflammatory factors in the circulating plasma were trending down. And also, then there's another human pilot trial was reported in eBioMedicine as well. First time to reported that senolytics can decrease senescence markers in human tissues, in adipose tissue and in the skin biopsies. This first study, reported senolytics decrease senescence markers in humans was also published from my mentor's group, Dr. Kirkland's group at Mayo Clinic, was at Mayo Clinic. And then also another trial now is testing in patients with Alzheimer's diseases. And they also published those Alzheimer's patients were well tolerated with senolytics. Now we're looking forward to see the phase 2 results, the efficacy of the senolytics D plus Q. And with the success of the pilot trials and preclinical testing, there are numerous formal trials under the way. I wouldn't introduce each one, but I listed them in this table. You can see how many they are. So they're under the way. We are looking forward to see the efficacies or their effects in those diseases. And now I want to talk about more the rationale why my research are focusing on the role of cellular senescence in aging and diseases. First of all, I want to talk about cellular senescence, my link to other aging pillars, because for a long time that I have a puzzle. We know that senescent cells are actually a rare population. Even with aging or disease, you only can maximum identify like 5 to 10%. So with such a rare population, even you target a subpopulation of those senescent cells, you can get a disproportionately large beneficial effects. Why is that? So then we speculate that the cellular senescence might link to other aging pillars, which means when you target cellular senescence, you also target other aging pillars, which can lead to a bigger beneficial effects. So the first aging pillar I was thinking about, it was alpha clausel, because for a long time that alpha clausel being discovered uh, like decades ago, because people found that alpha clausel knockout mice, they display as accelerated aging phenotypes, and also when you overexpress alpha clausel, uh, it can it can increase healthy lifespan in mice as well. Not only in mice, in humans as well. That's why the name of this protein being named by one of the botanists who can actually spin the thread of human life. But anyway, in humans, it's been also well established. Alpha clausal expression are linked to all-cause mortality in humans. So then I noticed that with aging, there is accumulation or increase of senescent cells, however, accompanied with decline alpha causal. Maybe these two are linked. So to test the hypothesis, we first to test out in our in vitro assay. So we cultured healthy normal endothelial cells, like kidney endothelial cells were HUVECs, and also brain astrocytes with conditioned media from senescent cells versus non-senescent cells. We indeed found senescent cells can significantly decrease alpha clausal expression across 3 different cell types. And when we use normalizing antibodies to against each SASP component, we also found that the antibody against activin A or the antibody acting against alpha and alpha can partially restore alpha clausal. So, and also recombinant activin A or L1 alpha can also significantly reduce alpha clausal expression in those normal cells. So from these results, we know that the senescent cells could decrease alpha clausal through paracrine effects, and also to the different cell types, they may have different SASP component to decrease alpha clausal. So this is also gave us a rationale why we should target the senescent cells instead of target each individual different SASP factors. By targeting cells, we basically target those factors as a group. And then we also tested whether senescent cells can decrease alpha clausal in vivo. So we transplanted senescent cells in healthy mice, and indeed we found in urine and in the brain cerebellum and plexus, we see significant reduction of alpha clausal in the senescent cell transplanted mice. And then we ask if we target senescent cells, maybe we can restore alpha clausal. Indeed, the first proof of the concept study where we use this intact mice, where we can so the mice can we can treat them with AP201A7 to induce apoptosis, specifically in P16 high cells that we found with the treatment in the kidney and in urine and in plexus, hippocampus, we all see alpha clausal partially restored by AP201A7. So not only in transgenic mouse model, and also by Senolytics, we saw the same thing. Basically, the Senolytics effects pretty much recapitulate the transgenic mouse model, the results. So as you can see here, after treatment, D plus Q were fisetin, and fisetin is another senolytics that we published a few years ago to target senescent endothelial cells. So with these two different senolytics, we see increase of alpha clausel in urine and also in old mice, in diet-induced obese mice, and also in senescent cell transplant mice, we in 3 different models we see increase of alpha clausel in the urine, and also we confirmed the protein expression by Western blot in the kidney as well. And also we saw the increase of alpha clausel in cerebellum and coil plexus by the senolytics of D plus Q in mice as well. And then we also look into how senolytics change alpha clausel in humans. So as I mentioned earlier, that the first trial was tested the efficacy of D plus Q in IPF patients. Also in those patients, I measured alpha clausel in the urine. We indeed find D plus Q can significantly increase alpha clausel after treatment, as you can see here in humans. And also, not surprisingly, we found the SASP component in urine, they're uh, uh, inversely significantly associated with alpha clausel expression. So with these results, we are pretty much convinced that senescent cells are mechanistically linked to one of the aging pillar alpha clausel expression. And now I want to talk about how the role of senescent cells in the diseases. One of the disease I want to talk about is infectious disease. So as everybody know that the pandemic, the SARS-CoV-2 infection, this is pretty much also age-related diseases because the elder patients, they actually develop the worsening, the more severity of the infection than young healthy people. Why is that? We also speculated senescent cells might play a role there. So to test out the hypothesis, first we tested in the proof of the concept study. Again, in intact mice, we specifically induce apoptosis in P16 high senescent cells. And after the treatment, we see the mice after infection, the survival was significantly improved compared to the vehicle treated. And not only in P16, not only in transgenic mouse model, also by Senolytics, the chronic treatment of the fisetin, we also saw significant improvement in their survival after the exposure of anemia of the infection. Not only in so Senolytics not only increased the survival of the mice, we also observed a significant reduction of the viral load in those mice. And because of time, I wouldn't go through every detailed data in this story, but basically I summarized our findings in this schematic view that we found that pre-existing senescent cells in elder mice or in old mice, they actually already significantly damage the surrounding tissues by secreting the inflammatory factors to create this local inflammation environment. And also we found that the SASP can increase viral entry proteins such as ACE2 or TMPRSS2 to further increase viral infection. And also the SASP decrease antiviral defense proteins, which are IFAD-M2 and 3. And also we found another interesting thing is that antibody was reduced by the SASP as well. So then after the senolytics treatment, we found that first of all, inflammation was attenuated significantly by Senolytics, and the viral entry proteins were reduced by the Senolytics, and uh, uh, so uh, so viral entry proteins were reduced by the Senolytics, and antiviral defense proteins were enhanced by Senolytics, and also the production of the antibodies was also improved by the Senolytics. And we published this story in Science with a collaboration with under my mentor Dr. Kirkland, and collaborated with Dr. Laura Niederhofer and Paul Robbins at the University of Minnesota. And then I will also want to talk about another role of senescent cells in neurological disorders. A few years ago, we noticed that senescent cells might mediate the obesity in all kinds of obesity-associated disorders. One of the disorder we look at is the anxiety. So to prove so again, to do the proof of the concept study, we gave AP201A7 in high-fat feed diet-induced obese mice. And then we also assessed their anxiety behavior by open field. As you can see here, the high-fat diet-induced obese mice showed significant impairment in their open field behavior, as you can see here. And after AP201A7 treatment, the anxiety behavior was significantly attenuated by clearance of senescent cells. And not only genetic manipulation, also Senolytics D plus Q, we found the similar pattern in DBDB mice, that the anxiety behavior was significantly improved by the Senolytics of D plus Q. And then we want to look into how senescent cells affect anxiety. So we basically isolated the brain from obese mice, and we then we did a single cell proteomics by using CyTOF. In this way, we can distinguish protein expression across different cell types. So we actually found that the Senolytics were the clearance of senescent cells can actually partially reverse the neuronal progenitor cell exhausted by obesity. And we believe this is not the only mechanism, not only one mechanism to explain this phenomena. There might be other mechanisms. So we continuing the investigating what exactly senescent cells do to affect the neuronal progenitor cell exhaustion and other neurological disorders in the brain. So with that, I summarize my past research. Basically, the discovery of Senolytics. We discovered a whole bunch of Senolytics, including the first generation dasatinib and quercetin, and also BCL2 family member inhibitors, innovative class. And then now my research is trying to focus on the role of cellular senescence in aging and diseases. So we're hoping that with more understanding the biology of cellular senescence, we can find more ways, more precise way to design Senolytics to target each disease, or is basically, we think that the Senolytics are disease specific and tissue specific. So there are a lot of more study we need to really do in the to find out the biology of cellular senescence. And then I want to acknowledge my lab members, my postdoc one, and my animal tag Kurt, and also my big thank to my mentor, Dr. James Kirkland, and Dr. Tamara Ticonia. Now they moved from Mayo to Cedars-Sinai, and the support from our aging center, Dr. Pignolo and Dr. Labraza and Dr. Kosa, and all the other collaborators, our long-term collaborators from University of Minnesota, Laura and Paul, and our funding resource from NIA and Evolution. I think now is questions, and I hope I can answer your questions.
Great. Thank you, Dr. Zhu. That's a wonderful talk. Fascinating research on cellular senescence and the use of senolytics and therapeutics and anti-aging and disease. We've got a lot of questions from a huge audience today. So in the interest of time, we'll go ahead and get right into those questions. I want to try to get as many of those answered as possible. The first one comes from Manjeet Singh. And Manjeet is stating that most senolytics from natural resources are really poorly absorbed by the GI tract, and also the microbiome, the internal microbiome may play an important role in their PK and PD. Do you have any thoughts on how to overcome these challenges, especially with antioxidants?
Yeah, I'm not an expert in drug formulation or drug delivery, but I can try my best. Hopefully, I can answer something here. I agree with this person's question about the GI absorption problems, and also we not my lab is not testing like PDPI, but other labs, I think they in their studies they already published the dynamics, observation efficacies of Novitaclex for the dasatinib and quercetin. I think is currently under testing in the trials. So we will see when they publish those data.
Great. Manjeet also had another good question that we've heard a lot about in the media lately. Industry and aging scientists are looking at plasma transfers from young people into older or aged people. Do you think that has any effect on cellular senescence, like in a good way?
I do. I think well, maybe something. I'm not saying that senescent cells could be reversed by this kind of rejuvenation way, but uh, I I think could be like stem cells, because we know that stem cell may have repairing functions. They might have something to do with senescent cells, but this is just my big guess, my speculation. Those questions are actually leaving to everybody in this field to figure out.
Yeah, yeah, I'm sure that's a big area of focus. Um, okay, so Adele Elisabetta is asking, senescence markers are similar also in cancer cells. I think that's the question. And could the role of alpha klotho also be important in cancer cell senescence?
Yeah, this is great question. Actually, to be honest, I think we should clarify the cancer senescent cells. I'm old school. So for me, cancer cells cannot be like really defined as a senescent cell. Senescent cell by definition, first they should have capability of they're like progenitor cells, though they should have like a well-type cellular program. But however, cancer cells kind of like auto programmed, so they can have senescence like. So that's my opinion only. But then I also think that there are a lot of commonly shared markers between cancer cells and senescent cells. They may have common roles in two different cellular states. But yeah, so based on the question about alpha clausel, I think alpha clausel in general is a growth factor. It could be beneficial uh, in aging, but I don't know how the growth factor, uh, I mean the role of of those growth factors in the cancer. So I think that question needs to leave the scientists working in both field cancer and senescence or aging to find out to really well define the role of senescence in aging, cellular senescence in cancer. There are many questions, puzzle things.
Mhm. Sounds great. And I think you also answered Adele's second question there. So I'll move on to the next person. The next person is Carmen Jeanette, and Carmen is asking, what is your opinion on senolytics as a vaccine, or as it a preventative measure for age-related diseases like prior to diagnosis?
Um, Well, I'm not MD. It's kind of risky for me to answer this question. First of all, I have to say this is just my own opinion. So vaccine has I personally believe it has some potentials in the future to treat age-related diseases, but we have to be super cautious, super careful because vaccine can change your immune system and irreversibly, I think. So there again, what the role of cellular senescence in immune system, this is still not quite understood yet.
Right,
So, but I think it has potential.
right, great. Appreciate that. The next question is from my good friend Lyndon Foster, based in California. Lyndon is asking a very intuitive question here, I think, and this question is very simple. So senescence is no longer considered a normal part of aging, but something that can be treated?
I wouldn't say it's not a normal part of aging. So again, as I introduced at the beginning of my talk, cellular senescence basically served as one of the aging hallmarks. So yeah, if you have aging, and I mean, like if people age, they do have accumulation of senescent cells. Yeah, so I do think they're tightly linked. But cellular senescence could happen independent of aging, could be induced by disease pathology or other ways like a chemotherapy. But that can be like a different program, could be different program.
Mhm.
So I think we need to gain more evidence, do more research to understand the difference of age-related senescent cells or age-dependent senescent cells, or whether they are different from disease-associated senescent cells.
Mhm. All righty, thank you for that answer. And Lyndon had a second question. I think you addressed this in your very first slides, but just really quickly, it, can you speak to why senescence happens in the first place? Like, is it because of telomere shortening, or what's the molecular mechanism behind it? And really, is killing the cells the only way to move forward to address age-related disease?
Yeah, so telomere shortening might be one of the trigger senescence program in the cells. Could be well, we definitely saw that in vitro, but in vivo it's kind of hard to really completely clearly to prove that. But we do think senescent cells can be induced by all kinds of stresses, damages, or triggers. It can be all kinds of like a telomere shortening, like DNA damage, genome instability, and the lysosome dysfunction, all kinds of ways. So I believe the upper stream can be different, but the downstream, when they become senescent state, they do share a lot of common features or the common functions. But in different tissues or different diseases, they might have very distinct roles. That's my opinion.
Mhm.
And yeah, so again, I have to say that even though I developed the Senolytics, but I don't think Senolytics is the only way to target senescent cells. To target senescent cells, you can do other ways by rejuvenating your immune system or by targeting other pillars. It could be very possible. And also by inhibiting SASP. There are certain ways before we know what cells we're targeting, like really crystal clear about the role of those senescent cells. I think uh, I think Senolytics. I do believe Senolytics target detrimental senescent cell, but I don't know. We should find out more. What other cells could be targeted, or uh, uh, by inhibiting senescent cell accumulation, it could be another different ways to
Mhm.
stop this process.
Great, thank you for that. And Serge is asking, I think, a really good question here. Senescent cells represent just a small fraction of cells of any organism, really. So how is it that such a small amount of cells over the course of an entire organism can drastically impact the overall body functionality?
Yeah, okay, this is great question. I also mentioned this. This is one of my research focus to really convince everybody that why we target a small portion of senescent cells, we can get such big beneficial effects. Uh, yeah, so I think it's like in aging, I mean like during aging process where when you teach you we're already predisposed to certain type of stress or damages, they're they're pretty much, I mean, vulnerable. So when you give one trigger, you may induce sequential events. So even though this is a very rare population, but maybe they have more important roles. Sometimes the population, I mean the percentage of the cells where the population may not be may not be matter. It could be the type of the senescent cell matter or the location. That's why now in the field, a lot of people put a lot of efforts to map senescent cells in the tissue, try to understand more.
Mm-hmm. Great, great. Thank you for that. Hannah Conan is asking, you mentioned in your presentation that antibody production is actually decreased by cellular senescence. Can you explain how you think this works?
Well, yeah, I don't know actually. This is the observation we observed in this study. I think it's worth of the following research in really investigating what exactly senescent cells do to reduce antibody load and what type cells being targeted by this Senolytics can improve this phenotype. So this is also unclear field to me yet.
Mm-hmm. Okay, and the second question from Hannah is, do the SASP target B cells? Is that maybe a mechanism?
Uh, do the SASP target B cells?
Yeah, SASP, do they target B cells? Maybe that causes the limited antibody production. I mean,
That's
that's just
very
a
possible. Yeah,
speculative thought there. Mhm.
yeah, that's very possible. It could be like senescent cell through paracrine effects to affect the normal B cells, or it could be B cells, I mean, themselves to become senescent
Mhm.
to have very, to show decline of their function.
Mhm.
I mean, both could be possible.
Right. Okay, great. Thank you for that. Gideon is asking, what is the relationship between aging and the length of telomere? I think I just brought that up a few minutes ago, but did you want to elaborate at all on that?
Uh, I wish I could do more, but I'm not a big expert in telomere shortening. So,
Right.
but I do think I do believe that telomere erosion or dysfunction in telomere closely tied to senescence, cellular senescence. Yeah.
Mhm. Great. Jun Woo Na is asking, is there any hormone difference in, I'm trying to paraphrase this, in obesity senolytic studies? So I guess maybe a hormonal difference between individuals who are healthy and have minimal senescence versus those that don't. And is there an obesity correlation? Have you ever studied that, or do you know anybody who has?
Yeah, in my lab we don't do I mean we don't investigate relation of senescent cells and hormone produce I mean production. But this is very interesting topic. I don't do it, but my colleagues or my close friends and he's expert in metabolic I mean in obesity related metabolic dysfunction field. He hypothesized that senescent cells might manipulate like those metabol I mean the metabolites to affect metabolism. Uh, it could be also similar to what happening to hormone. I think senescent cells may have some function on the hormone productions
Mhm.
because I do see other people. I cannot say their. I mean, I cannot talk about their unpublished data, but I do believe. I mean, this is
Mhm.
linked.
Mhm.
Yeah.
Yep, great. And Junwoo's second question, this looks like a good one as well. does senescence affect normal neurons directly, or is it that the immune system affects the ability of the neurons due to senescence? So senescence would actually have an indirect effect.
Yeah, this is also a question we are thinking. That's kind of question stick to my mind for a long time. I do think that senescent glial cells have direct effects on those neurons. But neuron being uh, with aging or disease such as Alzheimer's diseases, tons of publications show that the neuron themselves showed senescence phenotypes or senescence features. So they also, although neurons are post-mitotic cells, by definition, they shouldn't become senescent. But there are tons of study out there also show they have senescence hallmarks there. So really, we are looking forward to the outcomes from those people study, whether we can target senescent neurons, or we should target other type of senescent cells in the brain to indirectly affect neurons. This is also I want to find out.
Yeah, it's a good question. Okay, so we've got a few minutes left. We got time for a few more questions. If anybody else wants to type one into the chat box. I have a quick question for you, Dr. Ju. I noticed that there are a lot of over-the-counter supplements. A lot of them are flavonoids, and they advertise to be senolytics actually, and they claim to have a senolytic function and are anti-aging in nature. Do you believe that those actually work? Maybe you mentioned this in your talk, like you could go to the local grocery store and buy a supplement off the shelf, and it would have an effect on senescence, like maybe rid your body of senescent cells to a certain degree?
Oh, this is hard question for me. I'm still young enough. I'm not thinking about take those supplements by myself yet. I have to admit that. But I do see a lot of people taking anti-inflammatory supplements. They might
Yeah.
have effects, but what exactly effects they have, it's hard to say. So yeah, in mouse study, I only can say that in mouse study, we do see the polyphenol compound or flavonoid. They do have either SASP suppressing effects, or they could kill some type of senescent cells. Only in mice. In humans, I have to say data can tell.
Right. Okay. Thank you for that. One product, I think it's called quercetin or something like that, off the shelf, claims it rids the body of senescent cells. So I was just curious if you
Oh, be cautious on that. I don't think
know.
it's been well studied yet.
Maybe it's snake oil. Who knows? Okay, great. Well, it doesn't look like there's any more questions from the audience. We'll finish up just a few minutes early. I want to thank a number of people. First of all, the attendees for joining us. We had one of the largest turnouts ever for a BioTalk Tuesdays webinar. So thank you, attendees. I have to just quickly say where everybody was from, because we had a truly international audience today, all across the United States. I won't get into the individual states here. We also have individuals joining us from Belgium, Norway, Ireland, South Korea, Scotland, India, Hungary, Australia, Germany, Canada, of course, France, Colombia, Nigeria, Portugal, Mexico, Poland, Egypt, Italy, and the UK. Huge turnout. Thank you all for attending, especially those where it's early morning. We really appreciate it. Also, Dr. Ju and Jane Hong, we appreciate your attendance. Jane, we certainly appreciate GenoBioTX and Shanghai Model Organisms for co-hosting this webinar today. I think, Dr. Ju, you have a nice relationship with Shanghai Model Organisms, so I'm glad to hear that's going well. Thank you for a wonderful seminar. It's very exciting work, and congrats to you and your team. And Dr. Kirkland for everything y'all have done to impact the study of cellular senescence in Senolytics. And also, I just want to thank my colleague and good friend as well, Priscilla Hernandez from SinoBiogical. She is kind of the moderator and the host here and made sure all the technical aspects of the seminar went off without a glitch. So thank you, Priscilla, for that as well. And with that, I guess we will sign off, and until next time, thank you very much, Dr. Ju. Bye-bye.